रोहित।
यस मैम।
मेरी आवाज आ रही है ढंग से?
यस मैम, क्लियर।
ओके, जस्ट हैव अ लुक कि अगर भारद्वाज सर जैसे ही आए आप उनको को होस्ट ऐड कर लीजिएगा।
डन डन।
अच्छा, ही हैज ऑलरेडी एडेड। डॉक्टर अंकुर शर्मा विल ऑल्सो बी ज्वाइनिंग। एड हिम ऑल्सो।
डन मैम।
ओके।
प्रगति मैडम, जय श्री राम।
जय श्री राम सर।
और क्या हाल है? सब बढ़िया।
हाँ जी, सब सब बढ़िया। बहुत बहुत धन्यवाद, आपने ज्वाइन किया हमारा पहला हमारा प्रोग्राम आपने ज्वाइन कर लिया।
अरे थैंक यू डॉक्टर।
जी जी जी। सर 3-4 मिनट वेट कर लेते हैं, अभी भारद्वाज सर ने ज्वाइन नहीं किया है, पार्टिसिपेंट्स भी अभी ज़्यादा नहीं ज्वाइन किए हैं। नंदिनी।
Yes, ma'am.
और जस्ट मेक अ कॉल टू भारद्वाज सर, जस्ट सी लाइक व्हेन ही इज लाइकली टू ज्वाइन।
Okay, okay.
आप ही कर लेते डॉक्टर प्रगति, आपके पास नंबर नहीं है क्या?
सर है, मेरे पास भी है।
हाँ तो आप करिए।
ठीक है सर। सर ही इज ज्वाइनिंग, कर रहे हैं ज्वाइन। रोहित,
यस मैम।
भारद्वाज सर ने ज्वाइन कर लिया है, वो कह रहे हैं आप उन्हें अनम्यूट करिए प्लीज।
Yeah, he
डन
has
मैम डन।
done it.
जय श्री राम डॉक्टर साहब, जय श्री राम।
जय श्री राम। नमस्कार, नमस्ते मैडम।
नमस्ते सर। सर अभी 64 पार्टिसिपेंट्स हैं, 5 मिनट और वेट कर लेते हैं।
श्योर मैडम, श्योर, इट्स ए
जी सर।
प्लेजर।
जी सर।
आपके ग्रुप में बहुत ज्यादा पार्टिसिपेंट्स रहते हैं।
जी।
ओके मैडम, जब भी शुरू करेंगे बता दीजिए।
जी सर, श्योर सर।
थैंक यू।
जी।
मैम विल बी स्टार्टिंग एट 6:10। अटेंडेंस 6:10 पर ली जाएगी सारे कैंडिडेट्स की।
ओके। Nandini, I think we should start. It's 6:10.
Ma'am, am I visible and clearly audible?
Yes. Good.
Okay. So, greetings, everyone. A very warm welcome to all the respected dignitaries, our esteemed speaker, and enthusiastic participants for joining with us today. I am Nandini Sharma, co-founder Agriculture Valley Research Foundation, feeling truly honored to welcome you all to the first lecture session of Krishi Navyug 2026. Before we begin, let me briefly introduce Krishi Navyug. It is a 3-month agri internship come training program designed to bridge the gap between academic knowledge and practical agriculture exposure. Through this initiative, participants get the opportunity to learn from renowned scientists and experts, gain exposure to modern agriculture and innovation, developing professional research and career-oriented skills. This program is not just about learning, it is about building confidence, gaining clarity, and shaping the future leaders of agriculture. So now it is time to welcome our esteemed speaker for today's session. It is truly a moment of great honor and pride for me to introduce a personality whose work has not only contributed to science but has also transformed the lives of farmers. We are privileged to have with us Dr. Chellapilla Bharadwaj sir, an eminent scientist in the field of genetics and plant breeding, who completed his academic journey from prestigious institutions including Agriculture College Bapatla and JNKVV Jabalpur. Sir has been a student of distinction, receiving prestigious fellowships such as Coromandel Fellowship, AP Government Merit Fellowship, and the ICR Fellowship. With over 28 years of outstanding scientific experience, Sir is a breeder par excellence, especially dedicated to improving the livelihood of marginal and small chickpea farmers. He has developed 25 high-yielding, climate-resilient chickpea varieties, and remarkably, Sir is credited with developing the world's first marker-assisted bred chickpea varieties for drought and wilt resistance, bringing India to the forefront of genomic-assisted breeding. Sir has also published over 210 research papers in globally reputed journals like Nature and Nature Genetics, and has received multiple national and international awards for his exceptional contribution. Sir, your journey is not just inspiring, it is guiding path for all the young minds present here today. It is truly a great privilege for us to learn from a scientist of your stature. As a host, I am introducing you. So, this session is yours. Welcome, and we are grateful for your presence.
Thank you. Thank you so much, Nandini, for this beautiful introduction. Thank you, Pragati, for inviting me, and thank you, Ankar Bhai. So I should be sharing my screen. I'll share screen. Where does this go? Share. Hope all of you are able to see my screen.
Yes, sir.
Screen. Give me 1 sec before I make one small single person so that.
Yes, please, Daksha. आराम से.
ये क्यों? Nandini, can you help viewing? I want to just have one person viewing, but I don't know. It's not
रोहित
responding.
जी प्लीज आप गाइड करिए रोहित जी.
सर क्या इशू आ रहा है?
ये जो गैलरी मेरे बगल में आ रही है उसको मैं एक में करना चाहता हूँ. हाँ, हो गई. Thank you, and I want to pull this. Oh my God. What happened now? Somehow
What
it
happened,
moved
sir?
away from sharing to your this thing.
सर एक बार स्लाइड मूव कीजिए.
नहीं मुझे तो स्लाइड्स यहाँ दिखाई नहीं दे रहा है.
ओके.
Okay. My
Do
screen.
one
It
thing,
has
sir,
come.
stop.
Now it has come.
सर एक बार ना स्क्रीन शेयर आप बंद कर दीजिए एक बार.
Stop share. Okay.
Yes, sir.
Then again I reshare, right?
Yes, sir.
It's moving.
Okay, sir.
Good evening, everybody. Today, I should be speaking on genomic assisted technologies for crop yield resilience. I should be not just covering chickpea, but also the whole gambit of marker assisted breeding or genomic assisted breeding, which has revolutionized rice, maize, your groundnut, chickpeas, pearl millet, pigeon pea, and the sort of crops, mostly the agricultural crops. And before I start my PPT, I think I should be very much thankful to all my co authors and friends who have helped me develop this presentation. Dr. Neeraj, Manish Rurkiwal, who works in Reliance Industries, Manish Pandey from ICRISAT, Rajiv Varshne from Murdoch, Patel, my colleague from Dharwad, Gopal Krishna, rice breeder as well as head of genetics, Meenakshi Sundaram, Director, Indian Institute of Rice Research, Hyderabad, Dr. Tara Satyavati, Director, Indian Institute of Millet Research, Firoz, a prominent maize breeder, Hari Krishna and wheat, and Professor Chaturvedi Saab, who is currently the Director Research at RLB-CAU. And together, this beautiful synergesis of gap in various croplands could turn out. Now we are faced with a very big challenge that our water footprint, particularly in case of crop production, is very, very high. It's so high that 1 kg of corn costs you 900 liters of water. When you start looking at 1 kg of wheat, it is 1300 liters of water. 1 kg of soybean costs you 800 liters of water. You need to understand that the water footprint is very, very high. And how do we reduce it? In the more sense that the clarion call given by our Prime Minister, Honorable Priya Shri Modi Saab, who says more crop per drop. Now, with this, we are faced with a gambit of challenges. You need to produce more food, and this more food should come from less resources, less arable land, less water, less labor. It's true that the Indian agricultural scientists, Indian technocrats, and the Indian farmers could withstand and overcome Malthusian principles where we were ship to mouth to today a food surplus country. From 47 million tons, we have almost reached to 300 million tons, but this does not suffice. While we start moving in 2050 to more than a 2 billion people, and we need to produce this, how are we going to do that? The population is increasing, whereas arable land capita is greatly decreasing. And that's where the translational genomics for agriculture would come out into a major role. Now, it comes into a major role because it is a tool. You need to understand a tool does not supplement a person, it only aids a person. That's what it's rightly said. Technology aids a change, it is not the change in itself. So you need to take hold of the technology and use it for the advantage. The whole idea was even earlier, we were producing varieties, and as the first principle is to have a genetic diversity from which a breeder can carry out selections based on the principles of evaluated diversity by the gene bank specialist, and then the trait genetics as identified together with a physiologist coupled with a geneticist and a breeder and identifying it. That's fine. But then we do understand that there is a huge amount of G into E variables, particularly the E variables. And that's why you had the stability models that were actually working to reduce this environmental variability. Be it the Finlay and Wilkinson stability model, be it the genotypic or the phenotypic stability models. But in either of the cases, we were trying to estimate better the phenotype based on its genotype. But then we were using the environmental stability models, that is, testing in multiple environments. And of course, we have now advanced multiple environment stability models like the MGI, or the blues and the bloods. But however, our idea was to look at these genes which are locked up as a huge reserve in this genetic bank, and how do you actually develop the climate resilient varieties. Once the markers have come down, so that you could clearly identify the marker trait associations, a strong marker trait associations of the invisible but most important traits, then what we can do? Based on the marker, you can actually pull out the trait from the gene bank and then actually look how it is traveling through your plant populations by marker trait associations. In the foreground, you select for this particular trait of interest, while in the background, you keep your own plant idiotype of interest. Therefore, you get a final variety which is having your trait of interest from the germplasm, but has trait of agronomic interest from the recurrent parent or from the recurrent population. So you have a large gambit of genomic breeding approaches, which includes one side the marker-assisted selection, marker-assisted backcross breeding, marker-assisted recurrent selection, while on the other side you have the forward breeding, haplotype-based breeding, genomic selection, and to now speak of genome editing. So what are the 5 Gs of crop genetic improvement? One, you need to have the genome information. And this is one of the most important crop. If not, you just take out pictures, just like I've spoken in earlier group classes, it becomes a molecular photograph and nothing helps. So you need to have annotated genome sequences which speak that what are the genes which are working for a particular trait, how are they expressed, in which genome they are there. You require genome sequenced assemblies. Pulses were an orphan crop, but over the years, thanks to particularly Crisat, Professor Rajiv Varshne, Molber from Germany, that today they no longer are orphan crops. At least chickpea is no longer an orphan crop. You need to also have a good amount of germplasm. If you don't have a germplasm that is well characterized at both molecular and agronomic levels, it's of no use. Most importantly, the third one is genes, that you need to identify a gene. Identifying a gene by MTA using QTL is great, but then you need to clearly understand that this gene does the exact function that you are proposing as an hypothesis. So what you need to do, not only you need to identify the gene, but you need to functionally characterize the gene. Not only you need to functionally characterize the gene, once you have identified a gene and you have functionally characterized, you need to validate that yes, under the similar conditions, when this particular gene is present in a different genotype, it elicits the same response. Then once you have this information, using marker-assisted breeding or various other genomic breeding approaches, you can feed forward the breeding, coupling it with what we call as speed breeding techniques. Speed breeding is no breeding, but it is taking more than one generation per year. Let's say earlier we used to take through rapid generation advancement 2 to 3, here you're able to take 6 to 8 generations per year. So therefore you're cutting down all the cycle and then have a population that can directly go into selection processes. And a new tool, genome editing, gene editing, and mind you, epigenetics are going to play a major role in the next 30-40 years. Now we straight away come to our examples, and you do understand that bacterial blight is a major constraint in the production of rice. Well, it is spread across the nation and even across the rice growing tract in the world. It is caused by Xanthomonas oryzae, Pathova oryzae, a major problem in irrigated and low-lying ecologies where there is about 20 to 30% damage. In case of India, almost no state is spared from BB. The management of the disease is to breed for resistance. And mind you, there are not one or two. So there are 40 resistant genes which are governed by a single R gene hotspot. So there are about 22 pathotypes of this gene. Thanks to the cudgels taken by Dr. Sundaram at IIRR when he was at Cuttack, then Ramesh Sonti and Vishnupriya and CCMB, that they could actually tag this gene. And then they had a very high yielding variety like Samba Mashuri, BPT 5204, the pride of Andhra Pradesh, which is very high yielding but highly susceptible to bacterial blight. So you need to bring about bacterial blight resilience in this variety. And this group could identify SS1113, which is the donor. It's a Chaki type. It has been developed at PAU Ludhiana and is resistant to bacterial blight, but scores very low by the millers and therefore is not preferred by the farmers too. Now, what they basically did was looking at this gene, the XA21, XA5, XA13, which was present in SS1113. Therefore, they started trying to marker-assisted bred. They should have been the male. So both in Samba Masuri in case of Triguna, which is already resistant to GM bacterial plant hopper and white-backed plant hopper. They've used the linked markers for foreground selection and uses marker-assisted backcross breeding. And they could develop an improved Samba Mashuri, which has a very high yield of more than 5 tons per hectare with a low glycemic index of 50.99. And if you look, it has a very high bacterial blight resistance. And this success story of rice biotechnology in India serves as an excellent example, not only what genomic assisted breeding can do, but also in interinstitutional collaborations. It is already there in 2 lakh hectares occupied by the country, has been licensed to private seed companies, and is actually covering almost 1,000 Samba Mashuri farmers. Now the rice group, both at Cuttack, at IRI, at Hyderabad, they're working on various other traits. Bacterial blight we have seen. They're looking at blast, and already IRI has gone quite ahead in releasing what we call as basmati varieties with Pi 2, Pi 54, which are resistant to blast, to that of BPH 33T gene, to that of Galmid GM4 and GM8, phosphor use efficiency, PUP1 genes, salinity saltol, as well as adult plant salinity tolerant genes, drought QDTY 2.1 and 3.1, submergence sub1 gene, and as well as looking at genes. So I think you have recently heard of the paper of the genome editing work by Professor Vishwanath, and that itself stands as an extraordinary feat by the Indian scientists anywhere for the first time in the world. But then a lot of products are already in gambit. are already under cultivation using genomic assisted breeding. Similarly, this is the work of my teacher and my guru, Padam Shri Professor A.K. Singh, where he has combined bacterial blight resistant and basmati quality characters. Once this particular gene's loci was known, they developed a huge amount of mass derived basmati rice varieties PB1637, which has blast resistance, and then PB 1728, which is bacterial blight resistance, and now they are working on combining these two in basmati as well as non-basmati based varieties. Dr. Gopal, Dr. Prolay, and Ranjit are carrying out this work. Another is the herbicide trait tolerant rice genotype. In the foreground, you see that the whole of it is through mutation breeding by knocking out the air and bringing down the ALS mutant, which gives herbicide resistance to imazethapyr. And when imazethapyr was sprayed on both the crops, you can see that the non-GM herbicide tolerant rice totally is tolerant, whereas that of the susceptible line is totally. Now, this is what is doing a lot of things. Similarly, how do you increase the grain weight per spike under drought and heat stress condition? In case of wheat, great work by Dr. Hari Krishna and group. In case of wheat from IRI again, and now this variety is protected and released and doing wonders, because wheat and chickpea both experience terminal drought and heat, and they have a line that has been basically released, and now they are looking at molecular mapping for yellow rust resistance in gene DBW43 to the new race so that this can be worked out. And they have a large amount of products which are actually being looked at how this particular marker scores when there is a very high temperature, and they could identify GWM261, which is linked to RH8 as well as having deficit moisture stress tolerance. Another excellent crop is pearl millet. The pearl millet genome sequence, recently a Nature paper has come, and they could actually sequence not only pearl millet, now they're sequencing all the millets. But as I said, having the genome information is the basic roadmap on which further inroads can be made. And then Professor Tara and her group, when she was at IRI, later when she moved as project coordinator for ACREP on pearl millet, now as director Indian Institute of Millet Research, have done an yeoman work in mapping QTLs that control iron and zinc, and exploring for drought tolerance too. And I think anywhere in the world, the first crop to have a benchmark which is praised world over for iron and zinc, the minimum benchmark for iron and zinc, without which a variety can be released, is pearl millet, that has been widely appreciated. So they have also, not only that, the genomic assisted breeding tries to identify the wide heterotic pools which form the basis of heterosis. Now that's what we do when we were doing generally the Mahalanobis D square or the PC analysis, where we try to identify earlier through the morphological characterization and diversity analysis, the heterotic groups. But now since you have molecular information, so you can clearly identify which are the diverse heterotic groups and carry cross combinations. And one of the thing which has been done, again, this is from Hisar, that HHB 67, which could be developed using mass resist downy mildew, saving millions of crops and losses. In case of maize, you know that maize is a rich source of particularly vitamin A and others, and used mostly as a chicken feed. But then how do you increase the provitamin A content or the tryptophan content. So Dr. Firoz and his colleagues have done an excellent work releasing almost 15 maize hybrids which have very high, for example, Pusa Vivek QPM9 has a very high provitamin content of 8.15. If you look at Pusa HM4, which is hybrid maize 4, which is again improved for tryptophan 0.91, lysine 3.62, and also maintaining very high grain yield of almost 64 quintals per hectare with a potential yield of 85 quintals, 8.5 tons, and then has almost a duration of 87 for the 75% dry husk. Similarly, if you are looking at HM8, again having much more of 1.06 and 4.18 of tryptophan as well as lysine. They have produced a large number, as I said, 15 varieties, but you need to understand that pre-breeding is one of the sources which actually plays an important role in making breeding more insulated for various abiotic resilience stresses or for increase in plateau. So what we need to have is a good germplasm resources, which I was saying earlier, that you need to collect these germplasm resources if they are not available or create through by either mutation or hybridization techniques. You need to identify the new genes and then transfer them into backgrounds which help in utilization in crop improvement program. In case of chickpea, whether you start looking at heat, drought, frost, cold, and salinity, key diseases to various insect pests, machine harvestability, extra short duration, quality, and nutritional trait. The germplasm serves an active resource, but how do you pinpoint the germplasm which carries this and actually the gene in that germplasm which carries this? So we have a different approach than just evaluating the germplasm all. So what we go as focused intensive germplasm screening, where the germplasms are screened at a hotspot and then you collaborate the data that has been obtained, and then you also at the simultaneously evaluate the molecular data and try to identify donor sets. And that's what we have done when we were actually looking into how much the Indian subcontinent chickpea was far away from that of the Ghana, West Asia, and North Africa. And being most divergent, we have started the pre-breeding program. And one of the beautiful things that we identified was that there has been a decrease in the genes which were selective genes for crop improvement from that of wild races to that of land resources. And to tap that, you need to look towards the land races and wherever possible the wild species. So while we were deciphering the 3000 chickpea genome sequencing initiative with ICRISAT, Professor Rajiv Varshne leading the group, you could actually create a genome variation map across a pan genome, identify the origin and migration routes, and how chickpea as a species diversified and what are the genetic load or burden which has become responsible for crop performance per se in case of chickpea and most of the pulse crops, so that you can make a basic foundation for genomic prediction and optimal contributions. And in this process, we could identify a QTL hotspot on linkage group 4 in chickpea. We actually using the marker assisted breeding technologies developed and introgress into a group of lines and release the world's first Pusa chickpea 10216. In the most driest regions, as I said, of having just 20 to third less than 20% at the time of flowering and harvesting, it still yields 1.5 tons per hectare, where other chickpea crops cannot come out, while under normal conditions, it gives 2.5 tons. Fitting excellently, making it a global sensation when it was released, that it has been touted as chickpea delivers super chickpea in record time, thanks that Professor Varshne's initiative and our commitment together. Now, the robustness of this QTL hotspot is not just in our country. Across the globe, which had the similar climatological conditions, whether it was Giletu, which was developed by my friend Asnan Fakare in Ethiopia, and that of what you call as Kenya, which will be coming along. And at the same time, we developed Super Anigiri, one which has AQTL transferred into the most popular variety by Dr. Mannur, late Sri Mannur, who is no longer there, into Anigiri 1 and making it as a Super Anigiri having resistance to wilt. Now, if you look whether by Paul Kimamoto, my friend from Kenya, or Asnan Fakare, or our own chickpea crops, we now have a large amount of marker assisted backcross introgress lines carrying this particular QTL. We have released PUSA 10216, PUSA 4005 from IIPR Kanpur L4-14, and of late, very latest PUSA JG16, which are highly tolerant to drought and terminal heat. While the marker-assisted backcross breeding could bring out Super Anigiri, Pusa Shraddha, and Pusa Chickpea Manav, which is counted as the giant Manav, which yields almost 4 tons, 40 quintals prati hectare, and having all the fog genes that have been pyramided. Similarly, now we are working on salinity, where we have screened a large amount of germplasms. We could identify a good amount of genes which are actually upregulated during what you call as the salinity stress. We could actually identify the differentially expressed genes. My friend, my student, and now my colleague, Dr. Neeraj Kumar, is working on them. And we could actually identify that the HKT1 plays a major role apart from TPK gene that plays in transportation as well as selective vacuolization of the salinity gene. While working on the incentivizing research in agriculture funded by Ministry of Finance, IRI, IIPR, CSSRI, Karnal, as well as PAU Ludhiana, we could actually introgress from the wild, particularly from the Pinnatifidum, the hitherto non-existing botrytis grey mold resistance. Not only that, this is the first variety which has been released for cultivation, PBG8 GL130482, which has been developed by Cicer pinnatifidum cross Cicer aratinum. And then this particular line shows not only highly resistance to BGM, but gives an higher yield and will become a boon for the Northeast farmers. Not only that, we could identify very specific QTLs that stand constant across locations and across years. One of the biggest trade that plays a major role for doubling up or tripling of farmers' income is bold seed size. And then we could, for example, tag this trait and then introgress. And today we have produced a large number of lines which were using what we call as the allele that we transferred from PG0515 into Goxi, which is very creamy, having durable wilt resistance as well as a bit tolerance to drought. So we could get large amount of Kabuli lines whose 100 seed weight is from 60 to 75, and we have now seeds, and we have the first time we have released the double dollar chickpea variety, which has about 60 grams, which we say unclassified seed or unsorted seed, while the farmers take 70 to 80 grams 100 seed weight, and it actually yields 2 tons per hectare, much higher than dollar variety, which gives 850 50 kgs and more than that. It has a whooping market price as on today at 12,000 rupees a quintal. Similarly, in case of groundnut, Manish Pandey and group, they started looking at increasing the oleic acid, one of the most important thing, and then they could develop three varieties: ICGV 15064, 15005, and 16005 through marker assisted breeding, keeping the oleic acid QTL in consideration. And for the first time, two varieties, Girnar 4 and Girnar 5, have been released. And further, they started moving at looking at how do they improve the tolerance and resistance to rust and leaf spot. And they're actually now undergoing the marker assisted breeding final cycle, and they're into the accurate testing. In case of a pigeon pea, a real tough crop that can be handled, the marker assisted trait varieties are looking at yield related traits for reducing the flowering duration, reducing the days to maturity, increasing the pods per plant, 100 seed weight, bolder and shinier seeds, seeds per pod, and increasing more number of primary branches. At the same time, looking at resistance to SMD and Fusarium wilt. So, a good amount of pigeon pea genome information has been identified. Large number of these traits have been what you call as tagged and no marker assisted breeding program, both for reduced short height, for increased branching, for increased resistance is underway. And the whole idea, while I was saying of this, is how are you increasing the breeding value of a genotype? Using marker assisted breeding coupled with speed breeding so that you can take more cycles, that is more generations per year, because there is cheaper to genotype costing. You can make selections off target years in a greenhouse or in a speed breeding chamber. You need to just cull out the unwanted ones. You can maintain a high intensity of the favorable alleles following the genomic selection, and then you can have large number of selections. So what does this breeding paradigm now indicate? While the traditional breeding took 10 years, the genomic assisted breeding could take, including release of variety, in max 6 years with 4 years of testing. The rapid generation advancement, when coupled with speed breeding, is able to make the breeder efficient by handling more number of process and then selecting very few which he can handle easily. So there are markers for complex traits, and as the complexity and the definability of markers is increasing, their ability to pinpoint which are the genes or genomic loci that actually govern these genes are increasing. So there are very n number of ways to increase response to selection. One is increasing variation, increasing precision, selecting harder, that means rejecting many, and reducing time. Now you have excellent genomic selection while takes care of first three. The rapid generation advancement or speed breeding, when coupled with your statistical tools like the sparse testing tool in the advanced evaluation nurseries, the blues and blubs have out of season nurseries where you can try. So as I said, rapid generation advancement is one way. The second is genomic selection, where the nuts and bolts are that you have a particular trait of interest, be it yield, be it a particular resistance. You have the founder populations, which are mostly the breeding lines. And using the base selection, you identify the parents of these breeding lines. So you do a first generation of intercross, and then what you do? you rapidly advance the successive generations by using a genomic selection which takes into gambit the rare alleles responsible for giving tolerance, which are included in the base population, as well as the common alleles which are present in the high yielding varieties. Therefore, you get a new germplasm as well as new varieties. So that's what IRI, ICRISAT, and Cornell University could do. And we could get a multiple sequence testing with very high prediction accuracy, which we are doing. Now, coming to the last one where genome editing is playing a major role, and congratulations to all the Indian scientists, whether it is Shenmukh Sundaram or his group and Professor Vishnath and a group. We have come with the world's first rice genome edited lines. We have a huge amount of progresses with NIPGR looking at nutritional importance of Indian mustard, and my own student, Dr. Lakshmi, who is working for genome editing of ALS gene in case of chickpea, with JU working on groundnut GE for high oleic acid, NIPB working on a large amount of crops from potato to that of banana to that of rice to that of IRI New Delhi, again working on rice and other group of genes. ICGEB on working on redesigning plant types to that of TNA, which is working on TGMS system using genome edited, or to that of Mohali, which is working under the guidance of Professor Ashwini Parekh on GABA rich banana, GE beta carotene rich banana, to that of again NBRI, Lucknow working on GE tomato, cotton, chickpea, rice, and brassica. So whenever genome editing work was done, which was done as I have indicated by Chinnuswamy and group, they could develop the mutants of drought and salt tolerant genes, which encodes a particular finger millet. These, zinc finger millet transcription factor. These particular mutants not only produced higher yield, but also had both under non-stress and down-stress conditions, but under stress conditions, they showed extreme tolerance compared to their wild types in salinity. So when I come to the conclusion, my friends, in brief, I would like to put that next generation genomic approaches are very useful for developing climate change ready crops. For a given crop, there is a need for any crop to develop genomic resources, genetic maps, genome sequence assemblies, germplasm collections, breeding populations, training populations are very important and need to be precisely phenotyped for traits of interest. Genome-wide association studies, mapping of QTLs that can pinpointly identify, and further validation and characterization of these genes becomes paramount importance. Once this information of the top 4 is ready, mind you, that's not a small challenge. Then comes the advanced or genomic assisted breeding technologies. And further, we are going for genomic selection, genomic editing approaches. I'm very much thankful, Dr. Pragati, Dr. Ankur, and for the excellent introduction Nanda had given. To the Director General, DDG Crop Science, Assistant Director General, Seed, Director ICRIRI, Srinivas Rao Garu, whole of my chickpea group for the scientists, and my co-author on this presentation, Dr. Neeraj Kumar, Dr. Rajesh, students and research fellows, project staff, technical staff, chickpea team, and my funding collaborators. Actually, I represent the work of all my students and research fellows who are here, who actually do the work, and I'm their voice. I think that's the final slide. I thank you, Pragati. I thank you, Ankar. I thank you, Agricultural Valley.
Thank you so much,
Thank
sir.
you all. I should stop share and open it for discussion.
Rohit, kindly open the chat box for questioning.
Oh, no chat. I think everyone were busy listening. I don't have. Please ask questions or let us discuss.
If anyone is having any kind of doubt, you all can write your question, or you all can directly unmute one by one and directly you can ask your question. अगर किसी को कोई भी डाउट है तो आप लोग अनम्यूट कर सकते हैं और अपनी क्वेरी सर के साथ डायरेक्टली शेयर कर सकते हैं. एंड इफ यू आर अनेबल टू स्पीक यू कैन टाइप योर मैसेज और मैं सर तक वो फॉरवर्ड कर दूंगी.
No, no, I can see I have opened the chat box also. Yeah, they can message you.
Yes, sir.
The session was itself, sir, self-explanatory. I enjoyed it a lot, and I got a lot of information regarding. I am not of genetics and plant breeding domain, but I really loved your session. And as I said, people are appreciating it.
Students and जितने भी participants हैं, please don't feel hesitant. Any sort of question or query that you have in your mind, you are free to ask. Any sort of question you have in your mind after you have listened to the presentation.
A huge number of participants, 106.
Yes, sir. Anyone else? Any one of you? You are free to ask. This session is for you people only. You are free to ask. Don't feel hesitant.
Someone has asked, can you explain more about the process of selection of marker assisted genes? Since I was basically speaking on a gambit of how it is applied, avoid it, but certainly it's not so difficult. What you need to understand is marker is what? Marker is a trait in essence, but basically a DNA fragment which helps you to visualize with the trait that is tightly linked, or to a gene that it tightly linked. So two things basically are required, that you need to have marker trait associations first identified. That means you require, generally if it is a major gene, we may not require unless you are going for a pyramiding, but minor gene where you would like to bring them into a particular genotype, you need to have tight linkage. So marker trait associations are basically tight linkages between a marker that you can visualize, of course, in a gel, to that of a trait which you cannot visualize. And you need to either, for example, quantitatively actually look, and there can be a lot of gene to E interaction. Since the marker is at the DNA level, there is very less gene to E interaction. So of course, iterations are required. But then once such marker trait associations are identified, so when you cross a genotype, you do understand that 50% of the whole nucleus material comes from the mother and another 50% comes from the father. This is the cliche here. There are some mitochondrial, there are some plastid, and there are some plasmid genes which are directly inherited from the mother. Therefore, for the first time, we use donor as the male parent and we use the recipient into which you would like to bring as the female parent, and we cross, we get a F1. Now, this F1 will have 50% genome from the male and 50% from the female. And therefore, we need to look at our marker of interest, right? So our marker of interest, as well as we wanted to look at the genes which are actually resembling the female parent. Therefore, in a marker-assisted parent, initially we do polymorphic studies between the donor and the female parent. And then we identify such farmers. Let's say, for our simplicity, there are 4 chromosomes or 4 linkage groups, at least 20 markers per chromosome. So 80 markers which are spread evenly across the chromosome, which are polymorphic between the parents, and the marker of which it is tagged in the donor parent. And then we take that, all those heterozygous lines, and what we do, we back cross it. While we are back crossing, as I said, we want the maternal inheritance to remain from the female parent. We use it as the female and the F1 hybrid as male, and then we cross it. Therefore, you can understand 50% of the female and 25% from the F1. So BC1 F1 will have almost 75% from the female. And what we are interested, we are not interested in whole of the male genome. So we keep a track of the marker trait association, which is tightly linked to our character under initiation. So this is called as foreground. The foreground selection is carried for our traits of interest, while the background selection is carried to recover the female gene. So when you do next round of back crossing, then what happens? 87.5% theoretically comes from the female parent and remaining 12.5. So one more back crossing. So you almost cross 90%. This is the stage when you critically evaluate. And then since family character starts, you select individual plants, go for plant to progeny row method, or you go for a rapid generation advancement. You may not wait till the whole of the plant matures and cull out those plants which are neither carrying the recurrent parent genome or not carrying the resistant alleles. So you can easily speed breed across environments or within a controlled environment. That's how we can go. Which type of markers are useful? Gene-based markers are most useful. Among the markers, earlier we used to go for SSRs because in most labs we can do, but with the decreasing cost, SNP-based markers, SNP genotyping is very, very cheap. You needn't have a machine. You can outsource it with just, if already available, most of the things. For example, you have a Professor Varshney SNP array for chickpea, rice SNP array, which has been developed both by NIPGR and at IRI and at IIRR. Then you have a maize SNP array. Now you have a groundnut SNP array. So you needn't bother. How can genomic intervention help in reducing the chemical footprints in pulse cultivation? Excellent question, Shetty sir. Now, there is one particular pest which is very difficult to control, and it was arslan. It was basically the storage pest, isn't it? And now to control that, the copy genetic transformation was done by a protein arslan. Now we are looking that there is a huge amount of arslan proteins present in the wild which give resistance to bruchets. We can look into genome editing of this. Now, second, we don't have a control for Heliothrus. We have a good IPM package, but can we look at genome editing and then use crossover species or bridging species directly, those which cannot be crossed with Aritinum? Can we cross them with reticulatum and then transfer this character even if it is genome edited? Excellent. Someone, Shubha. Now, if you look at marker assisted bred Pusa chickpea manav, it covers almost 40 to 50% of the total area in Madhya Pradesh. The largest chickpea grown belt in the world, the largest chickpea producing belt in the world. 50 to 60% of this comes with the fork pyramided variety. What is that? Chickpea, Pusa chickpea manav BGM 2021. Rice, basmati rice. You just look almost 80 to 90% from marker assisted bred variety. The heat tolerant wheat is now making waves. PG05, the HTW series, which has been produced. So a large amount of area is coming up when farmers are identifying and understanding the importance of this yield resilient cross. The use of EMS mutagen and how its advantage and disadvantage is a different question altogether. I think you can ping me directly on my thing, I should say. But these are some Cinderella chemicals and Cinderella organisms, Arabidopsis, and similarly EMS. EMS can easily be prepared. The pH, which is most important for a mutagenic chemical, it remains very stable. Its half-life period is very long. You have to just take care of this thing. Then of course, disadvantage is it's carcinogenic. You need to carefully handle. But most important is it causes base transitions. So point mutations can easily, unlike deletions and duplications, it causes base transitions, which are one of the basic ways. For example, while Professor A.K. Singh, Gopal Krishna and group were working on developing herbicide tolerant variety, they haven't used any transgenic or genome editing protocol. Simple mutation to have the dormant ALS gene expressed. I think I've answered all the questions.
Any more questions, participants? You can ask, sir. Sir is answering each and every question of yours so patiently and with so peace. So you can ask without being hesitant. Please ask.
I think the queries are clear.
Yeah, even I
Okay.
think so.
We'll proceed the session. Sir, thank you so much, sir, for such an
Thank you.
enlightening and inspiring session. Your valuable insight and vast experience have truly enriched our understanding, and it has added great value to this learning journey. So, it is a privilege for all of us to learn from a scientist of your nature, and we are sincerely gratitude. We are sincerely showing you the gratitude, and we are grateful for your time, guidance, and for sharing your knowledge with us. Thank you so much.
Good night. Good day. Bye. Thank you. See you
Thank
all.
you, sir. Thank you, sir. Thank you, sir.
You all can leave.